So my job, after having given you that extraordinarily important information that you've already seen 67 times as it cycled through on the screen today, is to welcome you all. So welcome. And secondly, just say one or two things before we get to the main thing. So, we all know that Matt is an absolutely brilliant scientist and a fantastic investigator in areas that are of profound importance to human health. And we all know that. But I was trying to find out something a little bit more about Matt. And I know that Kerry's going to tell you some stuff before, but I thought that as a researcher, I should go into the internet and find things out about Matt because our lives are so sort of on the internet. So I did that. And it was immensely surprising to me. I spoke to some people and they said that Matt's quite a private person. There's not very much that you'll find out about him. He's quite quiet. So I went onto the internet and I was immensely surprised to find out that he's a former England centre-back and that he was captain of West Ham and played for Arsenal and has 21 caps and unfortunately only one goal for England, which is a problem. And then I kind of looked at it again and it was Matt Upton rather than Matt. And so it wasn't him. And so I as a researcher, you kind of persevere, don't you? And I know this is what Matt does with his sponges. You persevere. And I put the name in again and I found out that he was, in fact, a town in Worcester County, Massachusetts, with a population of 8,000 people exactly, which is kind of suspicious, really, if you think about that. But there are some very famous residents of Matt Upton, or Upton Ma, as they seem to have called it. There must have been a typo. So there was a man called Samuel Taft who knew George Washington, who had 22 children, which is very interesting. It was a source of. So the town was also very famous for making shoes, and had the world's largest women's hat factory at one point as well, which is, I think, really interesting. But of course, that wasn't Matt here, but that's his town, obviously his spiritual home. And I finally thought that I'd actually put his name in properly, so I did that. And you find out a few things through that. And I found his Twitter page. So I know this is Matt, because there was a picture of him on the front of it. In fact, the first picture of Matt on his Twitter page shows him with his close personal friend, Princess Anne. So, we're finding more about Matt here. So he obviously moves in these highly elevated circles. It was a really nice picture of you and Princess Anne, I must admit. So I then looked at what he was posting, and he's very big on retweeting rather than tweeting. So he's a kind of social influencer. So I think what we have here is a social influencer who moves in the highest possible circles. He describes himself as working in natural product discovery. So I imagine, and there's quite a lot of sponges going on, so I imagine what Matt's going to be talking about is his new cosmetics collection that he's been working on. And finally, in the kind of last sidebar of it all, there were three things that were there, which were flagged up as things that, if I was interested in Matt, I'd be interested as well. The first one was about COVID and long-term COVID. And obviously, we're very interested in that. The second one, I think, tells something about the kind of personality. It was about how dogs were really helpful to people who were homeless. And I think that's very sweet, really. So it's very good that you support those sorts of things as well. And the last one, and I think this is probably where we want to leave it with Matt, the last one was a deep look into the inner circle of Boris Johnson's number 10 establishment. So the fact is that, so we see what Matt is. He's a friend of Princess Anne. He's a confidant of Boris Johnson, and he's a fine cosmetician, probably. And at this point, what I want you to be asking me, of course, is whether I'm feeling all right, having channeled Boris Johnson in this sort of way. But what I'm going to do now is I'm going to hand on to Kerry, who's going to give you some real facts about Matt before we start off. So there you go, Kerry. Thank you.
Well, thank you very much. And welcome, everybody. Matt was born in Nottingham with his twin brother, Joe, which is about as far as you can get away from the sea while still being in the UK. But that changed shortly after when he moved to Aberdeen, age 4, and he lived in a small village north of the city. He spent his time there messing around on the beach with his brother and watching his dad, usually getting wet, trying to paddle out to sea on driftwood rafts. These are some of Matt's earliest memories and possibly helped form a deep interest in the sea. A family move brought Matt to the southwest, where he grew up living near Bath, until he left to go to university. Matt has many fond memories of family holidays to Dorset and the north coast of Devon, and maybe less fond memories of rainy holidays in North Wales. Biology was a passion at school, but microbiology didn't seem to feature on the curriculum. And this topic was much news to Matt when he was reading university prospectuses as he decided where to take his studies next. In a move partly designed to get Matt back to the coast and nearer some mountains, he signed up for a degree in microbiology at the University of Newcastle upon Tyne. This was at the time when HIV was first being recognised and reported. But antimicrobial resistance, or AMR as it's known, which is the focus of his current research, was really not widely discussed. His degree was followed by a PhD there, working with the eminent microbial taxonomist, Mike Goodfellow. His thesis focused on isolation of actinobacteria and investigation of their capacity to kill plant root pathogens. Matt is studying this group of bacteria again, but now he's looking at their activity against human pathogens. A change of direction and a return to Aberdeen followed his PhD when Matt took up a research post at the University of Aberdeen with Professor Hugh Pennington, author of the Pennington Report into the food poisoning outbreak in Lanarkshire in 1996. The work in Aberdeen helped Matt develop his interest in molecular biology and introduced him to the more medical aspects of microbiology. A series of other postdoctoral research projects took Matt to Liverpool, New Zealand, and Bristol, and a short stint in Antarctica. Through these projects, Matt continued to develop his molecular skills. He took up his first lectureship at the University of Manchester in 2001. His research there led to the formation of the diagnostic test company Spectromics, and it was also where he started his independent research in antibiotic discovery. I don't think Matt intended to stay in Manchester for 12 years when he arrived, but it gave him time to grow and establish his research group. Matt says he was not specifically looking for a job when he noticed the advert for the post he applied for at the University of Plymouth in 2012. The attraction for Matt in the advertised post was the request for someone active in molecular biology, bioinformatics, and medical microbiology, which all aligned with the research he was then conducting. The idea of living in the southwest by the sea was also a bit of a draw. He and his family love living here, and Matt's research has continued to thrive since he arrived. The first grant he secured at the University of Plymouth as a staff member was awarded in his first day in post, the result of discussions with the clinical team at Derived that started on his interview day. Matt's research group works in several areas related to AMR. He won't today be talking about the vaccine development work or the infection control projects, but these are very important aspects of his work on different fronts to address AMR. Matt's research has been funded by UK research councils, including BBSRC, MRC, and Innovate UK. And his research was key in securing a significant recent investment from the Wolfson Foundation to purchase new equipment. You will see that there is a strong theme of commercialization running through Matt's career to date, and he holds several granted patents in antibiotic discovery and microbial diagnostics. In his time as an independent researcher, Matt has seen commercialization of research become more widely acceptable in an academic setting. It's no longer seen by many as selling out or moving over to the dark side. Matt and I were introduced to each other in 2016 by a staff member in the School of Biomedical Sciences. Thanks, Simon. I was looking for a tame microbiologist to look at my deep sea sponges because I'd learned sponges carry interesting microbes that do things people benefit from, like recycling nutrients. And I wanted people to care about the deep sea, and this seemed a good way to show them why they should. Matt's motives were arguably more noble than mine. He was already interested in marine life as a possible source of new antibiotic compounds in the fight against AMR. So we got chatting and our collaboration began. And that collaboration has been a fruitful one. Our first joint PhD student successfully defended her thesis yesterday. Well done, Poppy. And we currently jointly supervise 3 other PhD students. I'm immensely grateful to Matt for agreeing to look at the deep sea sponges I had in the freezer 5 years ago. And I think Matt has secretly enjoyed crossing into the world of deep sea biology, and especially seeing strange and novel bacteria that he's been able to grow from the sponges. For me, working with Matt has been immensely valuable on a number of levels. He is an outstanding researcher and highly worthy of the title professor. He has a passion for tackling the threat posed by AMR. But he's also willing to cross disciplines in all sorts of things, including social sciences, arts, ocean governance, you name it, Matt's got involved in it with me at his side. He's very generous with his time and also the attention that his research attracts. I consider him not only a colleague, but a friend, and one who has helped me move closer to my own goals. So it is my very great pleasure and privilege to introduce Professor Matt Upton.
Well, what can I say? That was all very great to hear. Well, almost all very great to hear. Thank you, Shubi. Well done on the research that you've been doing. Thank you from me to everybody that's here, everybody that's online and tuned in and decided to come along and listen to me talk a little bit about my research in this field and give you a little bit of background to my history. I'd like to tell you a little bit about how I've come to be here today, about some of the decisions I've made along the way. And I'd also like to use this as an opportunity to thank a few individuals as we go through the talk. And also maybe coming back to Kerry's point about commercialization, talk to you a little bit about the commercialization and also a little bit about my kind of interest in technology. So, before I do that, what I'm going to do is embarrass my daughters who are here. And a big shout out to one particular daughter who's not here. Martha's on the left-hand side of your screen there. These are my amazing family. My wife, Faye, in the middle, and Ella, Kate, Martha, and Erica. Martha can't be here tonight. She was in Brighton Hospital having her appendix out on Saturday. So it's been a fairly mad few days. And I might come back to that a little bit later on. You're amazing. I love you all. And thank you for coming along. That's the hard part out of the way. So I thought it would be useful to give you an indication of what sort of things I'll be covering in the next 45 minutes or so. As I say, background to my education, maybe filling in on some of the information that Kerry's already started to give you, talk about my research posts, and then talk more about the independent research career that I've followed in antimicrobial resistance, or AMR. So at that point, partway through the talk, rather than right at the beginning, I'll introduce the topic of AMR and say a little bit about that. And I'll emphasize the need for new antibiotics and say what myself and Kerry and others are doing to try and find new antibiotics. So hopefully there's something in this talk for everybody who's here and online. Lots of friends, colleagues around the world, South Africa, New Zealand, Australia, Saudi Arabia, all tuned in, and I'm very grateful for you and doing so. And I would like to thank you for that and hopefully give something back. So even people that know me quite well might find out some new things about me through this talk and already obviously have done. I'm going to start with a question for you. So if you're in the room, can you please raise your hands? If you're online, can you please use the raise hand button on Zoom? So the first question from me is there anybody who has never used an antibiotic, never had a course of antibiotics? Nobody in the room. Okay, all right. And one person. And is there anybody in the room who, maybe one in the room who hasn't, anybody in the room who's had a course of antibiotics and then because those antibiotics haven't worked, has had to take a second course of antibiotics? Maybe half a dozen or so here. So a few. Going up. 14, 50 out of roughly how many? 90, okay. So fairly small percentage of people who have never had to take a second antibiotic. And I think what I'm hoping to do there is something I'll come back to later. Clearly, I'm trying to demonstrate that antibiotics are relevant to everybody. We've all taken pretty much a course of antibiotics. So I'll come back to that later. But what I'm going to do before that is talk to you a little bit about my background in some of the posts that Kerry's already mentioned, bringing us to the bottom left-hand corner to my research in Plymouth. And as Kerry said, I've taken a bit of a winding route on the way to where we are today. I'm going to start with a picture from my A-level biology field course at Dale Fort in Wales. This is my entire A-level biology class, including my twin brother, who's, I'm sorry, Joe, I've cut your head off at the side there. That's just my brother, Claire Rugg. Claire's online, I think. Claire and I have actually started doing some research together recently. Claire is, she's head of conservation at Paignton Zoo. She's also taught marine ecology and conservation. I'll come back to that in a moment, and myself here, clearly. So this trip, I think, was an influence on me in getting interested in marine ecology. Not deep sea yet, but there we go. And clearly had an influence on Claire. My brother's an oceanographer, so I think there's a bit of a theme there. The other thing you can maybe notice is a bit of an 80s Mel Gibson mullet going on there. Sorry about that. So I was interested in marine biology, but didn't think of studying at a university. I went to Newcastle University and I studied microbiology. This is the class of '89 on my graduation day. Hair's getting a little tidier. When I wasn't studying microbiology at university, I was mostly hanging out with the canoe club, and that's where I met Faye, my wife. So I'm kind of grateful to the canoe club for that, if nothing else. As you've heard, I stayed in Newcastle for my PhD, and I worked with Mike Goodfellow. Mike Goodfellow has pretty much written every single book there is, I think, about this group of bacteria called the Actinomycetes. You may not have heard the name before, but I think you're probably more familiar than you think you are with this group of bacteria. I don't know if you know, when rain lands on sort of fresh soil, there's that classic smell of rain on soil. That's chemicals produced by these sorts of bacteria called geosmins. So you do know them even if you didn't know it. This is my PhD summarized in 5 pictures. Basically, I grew some bacteria on the left-hand side. I looked at them down a microscope to see what sort of bacteria they were, and I tested them to see if they would kill fungi that infect the roots of plants. That's essentially my PhD summed up. Working with Mike was amazing. He's, as I say, an incredible knowledge on this topic. He's still practicing his research in microbiology at the University of Newcastle, and we've spoken recently. But basically, I did manage to find some bacteria living in soil which could kill plant root pathogens, and they had potential for biocontrol. I was doing my PhD. It was an industrial collaboration based at Sittingbourne Research Facility for some of my time. So this was my first kind of exposure to commercial research, and I think that's been something that's been with me all the way through. But I then became more interested in the medical aspects of microbiology and traveled to Aberdeen, as you've heard, to work with Hugh Pennington. One of the attractions was to work with molecular biology tools. And this machine just behind me on the screen, if you're familiar with this technology, this is an ABI 373 sequencer. At the time, it was all the rage. It was the first automated sequencer, and we were using the first automated DNA sequencer in Scotland. So I was very excited about that. But what we were doing was working with this group of bacteria called Streptococcus pyogenes. You might have come across this as the flesh-eating bug. And the other thing that happened when I was in Aberdeen working on this project is I was introduced to the media, the good and the bad of the media. So this is a story that came out in the Daily Sport, I think, about the project that we were carrying out. So this was me becoming interested in molecular biology and becoming exposed to the media. next of my postdoc travels, I went to the University of Liverpool, and I was quite keen to not spend my entire life in the lab and get out into these sorts of environments and do some environmental microbiology. So I took on this PhD, this postdoc project, again using molecular techniques to look at bacteria that live in these sorts of areas and produce methane. So we were using some molecular tools to try and identify what these bacteria were and see what they were doing. In the 2-year project, I managed to go into the field twice. So I kind of came to the realization that I'm going to spend most of my life in a lab as a microbiologist. One of the other things I managed to do while I was here was publish a paper with my dad, which is really quite cool. My parents have been supportive all the way through, but I think having a parent who can help you fill your CV with additional papers is a real bonus. So that was great for me. Thanks, Dad. And it was after this that I then took my first trip to New Zealand, and I started to work with Professor John Tagg. He's shown in the middle here holding a plate at the bottom of the photo if you can't see the pointer. John Tagg is another eminent scientist. He's a world leader in streptococcal biology. That's this sort of organism here that's shown in the middle picture. There's a few other characters on this image. Back row on the left is a person called Scott Beatson, who was an MSc student at the time, and I'll come back to Scott in a moment. So this project allowed me to start working again with these Streptococcus bacteria that I'd worked with in Aberdeen. And I was working in this building, microbiology building in Dunedin on the South Island of New Zealand. The only thing I knew about this city, Dunedin, before I went there was somebody in Liverpool had been here before and said they had great parties on the roof of this building every Friday afternoon. And I can kind of vouch for some of those parties, I think. So that was about all I knew. But I had a great time working with John and the team in Dunedin. And one of the other things I wanted to talk about was some of those kind of moments where you have a realization, you maybe call them a eureka moment in the lab, where you see something and you think, that's it, that solves the thing I've been looking for. And this was one of my first sort of eureka moments in the lab, working with this group of Streptococcus bacteria, producing a compound, an antibiotic called Salivarisin A. And it may not look very much, but on the far side of this image, there's a circle around a little blob on an autoradiograph. This is a photo from my lab book at the time. And this was a piece of data that allowed us to propose the model for self-regulation and production of an antibiotic by this bacterium. And at the time, this was really quite a surprising hypothesis that we were coming up with, but I'm not going to be able to go into the details today. So that was really quite a special moment in the lab. That was carried out in Bristol. I was working part of the New Zealand project in Bristol. I went back to New Zealand and picked up another project. And that's where I had my kind of second eureka moment, working with a related antibiotic called Salivarisin B. This was a project that had been going on for several years in the lab, and they were struggling to get the final piece of the jigsaw to solve this problem they were working with. So I took a bacterium called Streptococcus salivarius K12, I grew it up and produced some Salivarisin B and managed to generate some amino acid sequence data. That's information at the top there. What that allowed me to do was search it against a database of known proteins. And again, this doesn't look very exciting possibly, but this was a moment where I knew I was hopefully going to be able to solve the final piece of the jigsaw that they've been trying to solve. So this might mean something to some people in molecular biology, but basically I used this information to search out the full structure, the full sequence of the protein that we were trying to work with. This allowed us to submit a patent application, my first patent application, and it supported John Tagg being able to form a company called Bliss Technologies. This company is still active today. So that was very much a sort of my realization of biology of this group of antibiotics. It helped me understand the genetics of production of those antibiotics, and I'll come back to that in a little bit. And it was also clearly an introduction to commercialization of research, formation of a company to take research further. And it was kind of with that information in the back of fresh in my mind, I took up my first lectureship, my first independent position at the University of Manchester. That was in 2001, and I was pretty much immediately thrown into teaching and then running of the oldest postgraduate microbiology program in the UK at the time, possibly in the world. And I learned a huge amount of medical microbiology at that time. That's where I really kind of learned much of the medical microbiology I know today. And that's thanks to some of the people on this picture, particularly back right-hand side there is Dr. Noel Preston, who is an incredible microbiologist. So this also allowed me to set up my first research group, and I was studying their microbial genomics, which I'll say something about in a moment, and also natural product discovery. So this is when I started my real antibiotic discovery work, and I'll come back to that in a moment. This is a picture of the first grant that I was actually received as an independent researcher, the princely sum of £1,740. This allowed me to get started in the lab, though, and start building up my resources. I'm going to take a little sort of sidestep now, really, in some ways, and talk to you a little bit about some work that we were doing. And I want to use that. This isn't about antibiotic discovery, but this is me becoming more and more involved in understanding bacterial pathogens, how they cause disease and how they become resistant to antibiotics. And also about collaboration with clinicians. So we were working with a clinician in North Manchester who had spotted a really worrying trend. He was looking at these sorts of bacteria. These are called E. coli. They cause urinary tract infections and bloodstream infections. And he'd seen this increasing trend of bacteria that were associated with fatal infections and also had a range of resistances that you can see at the top side there. So they're resistant to 3 major classes of antibiotics. And to him, that was really quite worrying. He knew we were typing E. coli bacteria. So he got in touch and he asked us to type bacteria, these E. coli. And this was using the latest generation, a new type of sequencer. So it's again, me following that interest in technology. So we took 100 of his isolates and typed them using a new molecular biology typing method. And we found out they were of this special kind of E. coli, a clone of E. coli called ST131 E. coli. This had never been reported before. And we published a paper in 2008 reporting this clone of E. coli, this strain of E. coli. We were picked to the post. Our paper came out in March and somebody came out slightly before us in February. But 2008 was the year that this strain of E. coli became kind of more widely known. And we now know that this strain of E. coli, ST131, is the biggest cause of E. coli type disease that we've ever seen. We wanted to know more about this bacterium. So what we did is, again, using DNA sequencing, and we've now got to these really quite large and fancy machines. This is now called the next generation of sequencing technology. And we were now able to look at the whole genome of a bacterium. And that allowed us to look at things like genes for causing disease and genes for being resistant to antibiotics. And so we use this technology, and I'm now working with Scott Beatson, who's now associate professor of microbiology at University of Queensland. So we collaborated again together. He was from Otago. And we used this. I'm not expecting you to look at the detail of the figure on the right-hand side there, but if you notice, there's a red and orange and green colors here. These are certain strains of ST131 E. coli. And the ones in green were causing the biggest problems. And they were the ones that had been noticed by the clinician in Manchester previously. The other thing that Scott was able to do was show that this green group of this green strain of bacteria was able to kind of explode across the world and dominate across the world, causing these infections because of introduction of a quinolone antibiotic, so a certain type of antibiotic in 1986. So using this technology, we were able to understand how these bacteria spread around the world and what set them off. And we were able to show that that was linked to the use of quinolone, a certain antibiotic, in humans. So that was my understanding of kind of the role of human use of antibiotics in generating the problem that we're trying to address today of antimicrobial resistance. So, as I say, not directly linked to discovery, but it helped me in my understanding of the sort of pathogens that we're working with today. So back to these pills, to antibiotics. It's been suggested that the introduction of antibiotics has been the single most significant intervention in modern medical practice. It's known that they have probably saved 200 million lives since their introduction. And it's known that they probably added more than 20 years to human life expectancy. It's also now clear, as you're probably aware, that their use is threatened by antimicrobial resistance, or AMR. And it's also been suggested that if we don't do something to address AMR, they could result in infections that can't be treated that kill 10 million people a year by 2050. So if you put those numbers in the context of the current pandemic, we've just passed 5 million fatalities due to COVID-19 in 2 years. So 10 million a year is obviously a very significant burden of disease. I'm not sure if you're aware, but hopefully you are. Today marks the end of World Antimicrobial Awareness Week. If you work in the field, your social media stream has probably been full of information about this. Perhaps if you don't work in this field, it may not have been so full. World Antimicrobial Awareness Week is an initiative that is being followed by people in the field around the world. And there are messages of spreading awareness and stopping resistance. So trying to increase awareness of antimicrobial resistance, leading to change in behavior and change in medical practice to help address the problem of antimicrobial resistance. Come on to explain what that actually is in a moment. You may have also seen AMR described as a hidden or silent pandemic. And that's because it is a pandemic. These resistant infections are happening all around the world, but it's a pandemic that's happening very, very slowly. And I think because of that reason, it's not as widely recognized as it maybe should be. So to say something about what AMR actually is, in the simplest form, it's shown on the figure on the top right-hand side of your screen here. And it's something that happens naturally. So antibiotics target essential features or functions within bacterial cells. So if a bacterium randomly mutates, and this happens all the time, so that function can't be targeted by an antibiotic anymore, then the bacterium that's had the mutation is resistant to the antibiotic. And in that situation, the drugs don't work. The bacterium is not able to be treated. Usually that's not a problem. But if we use an antibiotic, that then kills all the bacteria that haven't mutated. So they then are wiped out, and leaving only the mutant-resistant bacteria. So they then flourish in circle 3 here, and they're now the dominant bacterium. And if you have an infection with a resistant bacterium, it can't be treated with antibiotics. The other thing that they can do is the mutated DNA can be transferred to a different bacterium. So the resistance is transferred to a new bacterium, and this new bacterium is then resistant to the antibiotic as well. Then what can happen is I can pick up this resistant bacterium, get on an airplane and fly to South America, and I've then taken that infection with me. So the resistance can transfer between bacteria, and we, international trade and travel, then transfer the bacteria around the world. So it's a global problem. And as I say, when you become infected with a bacterium that can't be treated with an antibiotic, clearly we can't treat infections. But I think what's less well known is that that also makes a lot of operations and our medical procedures makes them not possible, or makes them dangerous. So we've gone from a situation where antibiotics were kind of miracle cures, or almost novelties really in some ways. They were introduced at the time of the Second World War, and they saved probably hundreds of thousands of lives during the Second World War. And in less than 80 years, we've gone to a time where many of our modern medical practices would not be possible without antibiotics. As I mentioned earlier, Martha had an appendix out on Saturday in Brighton Hospital, and I spoke to one of the surgeons about that procedure. She had 2 days of intravenous metronidazole and the cephalosporin, 2 really important groups of antibiotic. If it was a nasty infection, she would have had an additional type of antibiotic. So I said to the surgeon, what would happen if you couldn't use those antibiotics? And he said they'd have to go ahead with the operation anyway, but they would just have to deal with the potential infection in as best a way as they could. So they might drain the infection and help the body try and fight the infection, but they would have to go ahead anyway. 1 in 20 people get appendicitis. And at the moment, it's a very routine, straightforward operation. But clearly, in a situation without antibiotics, it could be very different. So it is now really becoming acknowledged as a major problem. Most countries have these national action plans that set out the ways that we need to address antimicrobial resistance. It was recently discussed at the G7 meeting in Cornwall. It's been raised by the UN. It was a high-level meeting on antimicrobial resistance in 2016, and it's been discussed at places like the World Economic Forum and by the World Bank. On the topic of economics, I should probably really emphasize the fact that most of the places where AMR is going to have the biggest impact are the areas where resources are lower and antibiotics are not affordable. So I think there's a lot of work to do around, similar to the COVAX scheme with coronavirus vaccines being distributed around the world. We need similar systems for antibiotics to be accessible in all locations. And it's also led organizations like the CDC in the USA and the World Health Organization to come up with lists of bacteria that we need to target with investment and research if we're going to address the problem. There's a list on the left-hand side there. I've just pulled out 6 bacteria that we're working with in our research group that we're trying to develop new antibiotics for. I'd like to say a few words about this slide as well, because we're also becoming aware that this is a hugely complex problem, and we're not going to be able to deal with the problem just by looking at one particular area. AMR is relevant in human settings, in animal settings, and in the environment. And we need to think about that system at the level of the whole system to be able to address it properly. So just taking you through this image on the screen here, antibiotics, obvious what they are. We take them in the home, we take them in hospitals, we give them to our pets. We use them in agriculture. We're improving the way we do that, but we're getting better about using less in agriculture. And all of these antibiotics make it into the environment, and that's where resistance is selected for in the environment. So resistant organisms occur in the environment, they're acquired by us and cause infections. So if you try and deal with this problem in just one of these areas, you're less likely to succeed. And I'll come back to this right towards the end. One of those areas, obviously of interest to me, that we need to work in is finding new antibiotics. So I'll say a little bit about that now. You may be well aware of the story of Alexander Fleming over on the left-hand side there, credited with discovering penicillin in 1928, awarded the Nobel Prize for it in the 40s, along with Florey and Chain. So penicillin was introduced in the 1940s. Another person who was very active at the time, you may not be aware of, is this person here in the middle called Selman Waxman. He was also awarded a Nobel Prize. And that was for discovery of bacteria like these ones in the middle. This is another one of those actinomycete bacteria. And this produced a compound called streptomycin. And he was awarded the Nobel Prize for discovery of streptomycin. He also put in place a lot of methods that we actually use today for discovery of antibiotics. And it was an incredibly successful period, the golden age of discovery, where we discovered most of the antibiotics that we currently use in clinical medicine. So these were produced by bacteria that live in soil. And so Selman Waxman did a lot of work with soil, growing these sorts of bacteria and purifying the antibiotics they produce. We became less good at discovering antibiotics and changed to different technologies that I won't talk about now, but that basically led to the right-hand side of this figure here, a discovery void, where for 30 years we haven't been able to find new classes of antibiotics. So now there is a very urgent need for new antibiotics, and ideally they need to come from new classes. So I'll say something about a class of antibiotic next. On this slide, you can see 12 different classes of antibiotics. So these are sorts of antibiotics that work in certain ways or work against certain groups of bacteria. And resistance is now present to all of these different classes of antibiotics. So if I find a new tetracycline over here on the left-hand side, it's likely that resistance will occur to that new tetracycline very quickly because there's already resistance out there, and it's only a slight modification to that resistance mechanism that's needed. So ideally, we need antibiotics of new classes. The WHO carried out a survey, and they reported the results of the survey last year. They were looking at antibiotics in the development pathway. So antibiotics are being developed right from early stage discovery all the way through to use in the clinic. And their major conclusion doesn't make very good reading. So there are not enough new antibiotics in the clinical development pathline to meet patient needs worldwide. This is the WHO's take on things. So hopefully that leads you to think, well, so Matt's now going to tell us about what he's doing to find new antibiotics and new classes. And that's what I'll talk about in the remaining sections of my talk. The other thing about working with marine biologists is you can borrow really amazing photos like this of the sorts of places they go and the things they see. So this is a picture from a reef in South Africa taken by one of my collaborators, Rosie Dorrington, who I think is on the line. Hi, Rosie. So yes, what we want to do is go to weird and wonderful places to find weird and wonderful bacteria that produce antibiotics in new classes. Before I talk about the work specifically, I want to say something about why we're interested in marine environments particularly. So you've already heard the term natural products. I should have explained what that is earlier, really, probably. But natural products are chemicals that are produced by living organisms. And these chemicals have a really wide range of functions. And the marine environment has been an incredible source of natural products, so marine natural products, MNPs. And these have got all sorts of biological activities. The first marine-derived cancer drug was introduced 50 years ago. There are now many cancer drugs that have been found in marine organisms that are actually active and working. But there's also a source of other pharmaceuticals, enzymes for use in industrial biotechnology, the cosmetics that Shubi was maybe referring to earlier on, biofuels and lots of other things. And over 30,000 marine natural products have already been described, and more are being described all the time. So it suggested all this discovery, all this biotechnological potential could be worth over $6 billion by 2025. And I'd just like to say that I think that probably dwarfs some of the revenues that are suggested to be feasible from deep sea mining. It's also known now that a lot of these marine natural products, although they were originally thought to come from some of these weird animals up on the right-hand side of the screen, it's now known that they're produced by the bacteria that live in those animals. So these are microbially derived marine natural products, like the one on the right-hand side at the bottom. This is an antibiotic produced by, again, another actinobacteria that's been grown from a marine environment. So we feel this justifies us looking in the marine environments, and that's exactly what we're doing. So I'm really lucky to be able to work with Kerry, looking at some of these amazing samples. I've actually got some here. You can come and have a look at these later, maybe. But these are some of the samples that Kerry's retrieved from huge depths in the Atlantic Ocean and other areas, so more than a kilometre down on the sea floor, using these remotely operated vehicles, which have kind of opened up access to these sorts of environments in the last few years. So we feel this is a really valuable resource that we have here in Plymouth. There's only a few groups in the world that are working in this sort of area. I've already told you that deep sea, the sea has immense potential for biodiversity, and that we know sponges, these are some sponges that Kerry's taken photos of on her trips. These sponges produce products themselves, and they're home to bacteria that produce additional compounds, and it's those compounds that we're interested in. So what we hope, by looking in these samples, we will find highly novel bacteria that produce antibiotics of new classes, so antibiotics that work in new ways. This is a sample being retrieved from the deep sea by one of the ROV, remotely operated vehicle pilots. These samples are harvested from the seafloor, taken up onto the boat, and then brought to Plymouth, where we take over. And this is just a flow chart of what happens when the samples arrive with us. So that would be the sponge in the middle there. We were also working with additional marine samples, like on the far left-hand side at the top there. This is a seagrass sample that I took from Salcombe recently, having really quite positive early results coming from some of the seagrass. And that includes some of the work that I was doing with Claire, who was in the A-level biology field trip with me. This weird yellow organism up here on the right-hand side is a sea lemon, and this is an Antarctic invertebrate. We're working with these sorts of animals with the British Antarctic Survey. So what happens when we're given these animals, these samples, is we grow bacteria from them, we test them to see if they produce antimicrobial compounds, and then we follow one of two routes to try and find out what those antimicrobial compounds might be. We can take the bacteria and grow them in these flasks up on the left here, and then use equipment to purify the compounds out, and then try and characterize the compounds. Apologies, this image seems to be sideways. The other thing we can do is take these bacteria that we're interested in and look at their DNA. So do some DNA sequencing again. So we can analyze their genomes, potentially synthesize compounds that they might be able to make, and then again, test those in the lab and work out what those compounds are. The other innovation that we're doing, we're using more recently, is by cutting out this culture-based step in the middle. So not growing any bacteria, but directly sequencing the DNA. And we're doing this using the most recent innovations in DNA sequencing. And you may have got the impression, as I've been talking, of DNA sequencing technology, improving, machines getting bigger and faster and fancier. But the machine that we use now is shown on the bottom left-hand side there. And I've got one of them here. This is a MinION DNA sequencing device. And this produces just incredible amounts of data. You can almost use it in the field. But what we do is use it to analyze the DNA in these samples to look at potential new antibiotics without even growing the samples. So what I'll do now to demonstrate that in a little bit more detail is just take you through one example. And this is a marine bacterium that we grew from a sample like this, one of Kerry's. This is one of the first samples we looked at. And this bacterium is producing two potentially novel antibiotics. The first one of those we accessed, this is the bacterial strain. Looking down a microscope, it's got these really quite strange cells. We sequence the genome of the bacterium, and we can use software tools to analyze the DNA sequence and identify clusters of genes, so groups of genes that we think produce an antibiotic. So we were particularly interested in this one in green here, and we made a synthetic version of that compound, tested to see if it was active, if it could kill bacteria, and it could kill bacteria. So we then, we are confident that we'd predicted an antibiotic, tested it in the lab, and this strain produces a new antibiotic. The other way that we looked at this isolate was to grow it up in a flask, and as I said earlier, using these chromatography equipment, purify out the compound from everything else that's in there. We only want to see the antibiotic compound, so we purify it out using chromatography. And again, we tested that compound, showing it had antibiotic activity in the assay in the middle here. So we found a new antimicrobial active compound. Sorry, again, the image is flipped here, but what we did was characterize this compound using mass spectrometry. And I can't tell you what the molecular formula is. This is the kind of formula of the antibiotic compound. And I can't tell you what that is because we searched it against the databases shown in the middle, which is part of the procedure we follow. And there were no matches in the database, meaning this is a new compound. So we're keeping that information confidential for now. But what this allowed us to say is that this strain EU4 produces a new synthetic peptide antibiotic and a new natural product scaffold. So this is one example of a number of bacteria that we're progressing in this way. Some other highlights from this work are summarized on this slide here. And this is a sponge, again, a sort of sponge that Kerry's interested in, and it's from a group of sponges called the glass sponges, or hexax sponges that's shown here. And so for the first time, we looked at two different species within this group, Pheronema and Hertwiggia. So nobody had looked at these before for antimicrobial producing bacteria. So we did that, and we showed that Pheronema is probably the best of these sorts of sponges to be looking at, and we're now using that to inform our future search for novel antibiotics. We've grown about 3,000 bacteria from these different sponge samples and screened those bacteria for activity, and a number have been shown to be producing antibiotic compounds, and we're progressing those. But the other real benefit of some of these samples is, as I say, marine bacteria produce all sorts of bioactive compounds. So there's potential within this collection of bacteria to screen for anti-cancer compounds, pain-relieving compounds, compounds that improve our immune response, those sorts of things. And that's a real kind of bonus and extra value of these samples. The last project I'd like to mention on the marine discovery side is this really exciting new project that I'm carrying out with Rosie Dorrington from the Rhodes University in Makanda in South Africa. Rosie, I think, is on the line. Rosie and I were awarded some money recently to start this project, a 3-year project looking for novel marine natural products. With partners in South Africa and in the UK, and a commercial screening lab in South Africa. And although in the project proposal we stated that we will deliver new antibiotic candidates by 2023, we are confident we'll be able to do that. We feel this project has scope for much longer lasting benefit, and that's because we're establishing new capability both in the UK and South Africa for this sort of work, and supporting hopefully eventually the bioeconomy growth in both of these countries. So that's a great project, huge potential, and I'll maybe be able to report on that in years ahead. So hopefully that's giving you an idea of the work that we're doing in the marine discovery field. You may have the impression that some of this is kind of early stage. It's a long process that we go through in this work. So what I'd like to do to finish with is to talk to you about discovery of another natural product called Epidermisin NIL1, because we've progressed this now further through the pathway towards use in humans. So I discovered Epidermisin NIL1, I'll call it NIL1 from now on, being produced by a skin bacterium called Staph epidermidis. I started this work when I was in the University of Manchester, and I progressed that all the way through to today, the University of Plymouth, and that led to the formation partway along the line of a company called Amprologix. I'll tell you why we needed to form that company as we go through the next few slides. I also thought it was useful at this point to put in context the sort of the discovery part of the pathway to a new antibiotic, and that's shown on the bottom here. So Epidermisin NIL1 has been through the first 2 stages in this process. These are probably the longest stages. The clinical development stage in the middle here can be quite protracted, several years maybe, but approval and marketing hopefully afterwards would be a lot faster. The other thing I want to put on this or demonstrate on this slide is the number of compounds that you need to progress through this pathway to get one drug at the end. So it's suggested that 10,000 kind of candidate possible antibiotics right at the beginning gets filtered down as you go through this pathway. So we're now with NIR1 at this point here, at the end of the preclinical development phase, about to hopefully go into clinical phases and ideally into a new drug at the end of that. We're developing NIR1 to target this bacterium called methicillin-resistant Staph aureus, MRSA, you may have heard of, and its non-resistant version called methicillin-susceptible Staph aureus, or MSSA. MRSA is on the CDC list of priority pathogens. But just a few more bits of information about these bacteria. They both are still leading causes of healthcare-associated infections, so the sorts of infections that are acquired by spending lots of time in healthcare settings. And although there's been a lot of work in the early 2000s, 50% of bloodstream infections would have been caused by these sorts of bacteria. Lots of innovations in hygiene have brought the numbers down dramatically. But you can see for MRSA on the left-hand side of these two figures, the numbers are flatlined. For MSSA, numbers are actually climbing, and they're up probably about 40% on 10 years ago. The other thing to tell you about MRSA and MSSA is there's probably maybe 20-30% of the population are colonized with MSSA, and maybe 3% colonized with MRSA, the resistant version. If you're going to go into hospital for surgery and you colonize with these bacteria, there's a risk that you'll get an infection after surgery. And so one of the ways to deal with that is to decolonize you. And that involves shampoos for the hair and body washes, but also involves putting a drug up your nose to decolonize your nose, because MRSA and MSSA like to hang out in your nose. There's one compound in the UK that's licensed for this. That's mupirocin, shown on the right here at the bottom. And basically, that as a therapy is failing. Resistance is a problem for mupirocin, and we need something new to combat MRSA and MSSA. And that's what we're aiming to do with this compound Epidermisin NIR1. And that's because it's very, very potently active against methicillin-resistant and susceptible Staph aureus. So it's very potent and it kills these bacteria very, very rapidly, but it's also very low in toxicity. So you can take a dose 100 times the amount needed to kill the bacteria, and it's not toxic to human cells in the lab. We also, we've tried to make bacteria resistant to this compound, and we've not been able to in the lab so far. And we've done a range of other things that I'll take you through here. So this image in the middle, the crystal structure, this is the structure of the molecule. It's the structure it naturally adopts. And that's kind of great, you know, the structure. But this is the first structure of this class of antibiotic that's been seen. And we've used that structure to help understand the way the molecule kills bacteria. And that's shown on the left-hand side here. This is an image of a bacterial cell that's been targeted with the compound, and the compound's punching holes in the bacterial cell. We've also then taken the compound and used it to try and keep these caterpillars. This is an infection model. These are caterpillars we've infected with a bacterium and then treated with the antibiotic. And you can see the ones at the top that have been treated are healthy, and the ones at the bottom that have not been treated are not so healthy. So we know it works in this system, and it's has potential for use as an antibiotic in this sort of setting. But we've now taken it to the next stage. Shown on the right-hand side here is data from an infection model in the nose. So we've colonized animals in the nose and then treated them to remove the infection. And this is work that's required to be done to progress an antibiotic for use in humans, clearly. And really, to summarize what's shown in the figure on the right-hand side, a single dose, one dose of this compound was as effective as 6 doses of the mupirocin, the current standard of care. And this has not been seen in this model before, a single dose. And if this translates into use in humans, it could really have an impact on the way that we manage patients with MRSA and MSSA infections or colonization. So all of this was pointing it was all positive data, pointing in the direction that we needed to progress this towards clinical use. But to do that, really, we needed to form a company, and that's what we did. In 2018, we formed Amprologix from the university here to go through the preclinical phase of that development pathway. Shortly after we set the company up, we secured a government contract to carry out the work for preclinical development. And the first step in that process was making large amounts of this compound. And I mentioned earlier, Alexander Fleming, and he teamed up with Florian Chain, and they were awarded the Nobel Prize for introduction of penicillin. Florian Chain really, they were biochemists, and they turned Alexander Fleming's lab-based discovery into a medicine. Their work allowed it to be scaled up and produced as a medicine. And the equivalent for me, really, in a way, is a company called Ingenza, partners of ours throughout the preclinical and earlier development of Epidermisin. So really, in a way, Ingenza have done for Epidermisin what Florian Chain did for penicillin. So I am very, very grateful to Ian Fotheringham at Ingenza and the team that you have. They've taken us to a position where we've got a commercially viable compound that could become a drug. The other work that we did in this preclinical phase was develop a formulation, a final formulation, so a version of the drug that can go into the nose. We carried out toxicology analysis, and I'll talk to you about that on the next slide. So this is work, again, that's a requirement for progression of something towards a drug that you're going to put into humans. And this work is highly regulated and is carried out by CROs, commercial research organizations. So the formulation work, the NIL1 was formulated into a liquid. You can see it on the right-hand side here, sort of liquid that you could spray into the nose to dose a patient. And this was optimized for nasal delivery. The other thing they looked at was stability of the compound, because clearly you want something that's going to be stable over a long time. And stability was shown to be very good for 10 weeks. 10 weeks doesn't sound long, but because of the programs, because of the way they do this, they can project that out to be perhaps 10 months worth of stability, both in the fridge or at room temperature. So it doesn't need long-term storage at minus 20 degrees or other resource-intensive storage facilities. So this formulated compound, the active one is shown on the right-hand side here in yellow. That's the drug, and this is a placebo. So it's just a formulation without the drug. And we showed that the formulated compound was still active against things like MRSA, so that allowed us to go forwards. So another commercial research organization carried out tolerability and toxicology work. And so again, this is work that's required for progression. And they used a low, medium, and high dose in their assays, and they didn't see any toxic effects over 14 days. And all observations that they carried out, so looking for toxic effects in the assays that they were doing, no adverse effects at all. Everything was looking very, very good. So that puts us in a position now where we've got to the end of the preclinical phase, and Epidermisin NIL1 is now ready for progression into clinical trials in humans. And that's where we're heading, hopefully, for prevention of surgical site infections. And I just want to close by saying a couple of other things that we hope to do with this compound, because it's an incredibly active and flexible compound that we're trying to work with here. We know it kills the sorts of bacteria that cause skin infections like acnes, impetigo. So we wanted to develop it in this area as well. And we're starting to generate good preliminary data to help support that. Also, similarly with wound infections, we want to look at wound infections as well. And we've got some earlier preliminary data showing that we could also maybe use this compound to treat things like pneumonia. So this could be a pneumonia that develops in a patient following a virus infection or a primary pneumonia. And also we could use it in prosthetic joint infections. And this is another operation that requires large amounts of antibiotics to make the operation safe. So we think we could use Epidermisin in this format. The other thing we're trying to do with Epidermisin, and again, focusing on the crystal structure here, is make variants of it that work in different ways. And we're doing that by I won't go into the detail here, but this is a kind of a representation of all the different variations of epidermisin that there might be. So we're working with computational biologists using machine learning techniques to try and explore this kind of variability space to find new variants, new versions of epidermisin that work in different ways that we can take into different sorts of infections, opening up a whole new area of application. So that's just a kind of a walk through some of the discovery work that we're doing, some of the very early stage discovery work looking in the marine field, but also hopefully giving you an impression that we've got some things that have progressed much closer to the clinic and are developing towards use in humans. I mentioned earlier on, AMR is a very complicated system, understanding AMR and working to combat it, we need to look at the whole system and address it at many different levels. And so this is just summarizing some of the elements of research that I'm doing to try and address AMR in a One Health way. So that includes formation of the company and development of a candidate heading towards the clinic. These really amazing, exciting marine natural products that we're discovering and developing, and variations we're making of those using machine learning and artificial intelligence. We're working to raise awareness of the issue as well through public messaging and events and opinion pieces, those sorts of things. The U3A, I know there's a lot of U3A here and online, and I appreciate the way you've engaged with us and the work we're doing over the recent years. The final thing I'll say really in a way is sort of this in some ways is about a kind of a reflection of what I have been doing, but I'll also use this as an opportunity to maybe look ahead and look at some of the things that I would like to do with the work that we're doing and the work we're doing with Kerry, I think has been picked up by organizations interested in deep sea mining and other aspects of marine ecology and diversity. And so I'd like to be able to kind of inform some of those discussions as we go forwards. The final thing from me is to just thank additional people. I'm not going to go through all of these names here, but these are essentially the key people that I've worked with in the last sort of 20 years of my independent research career, people all across the world. And many people in the University of Plymouth, and the organizational teams and people and colleagues in the School of Biomedical Sciences, and a particular mention to my previous dean, Professor Rob Sneed, who was very supportive of me making my application for my promotion and helping me progress. The other people I'd like to thank, some of them are in the room. These are past and present members of my lab group. I've had many people come through the lab, and I've had a great time working with these people, and I'm honoured to be able to have worked with some of these people and help progress their projects. And that includes a number of PhD students, shown on the left-hand side here. As Kerry said earlier, Poppy Best made the jump from this group of current students to a group of kind of past students yesterday, and Matt Koch is kind of next in line. He's hoping to submit soon. So it's been a pleasure to be part of the kind of the journey of these students as they progress through their PhDs. Finally, really, I guess I've seen a number of these talks before, and they all seem to end with a sunset. So this is my kind of sunset photo. But it just remains for me, really, in a way to thank all of those who are here and online for their attention. And it's been a strange experience putting the talk together and reflecting on everything, and hopefully there's been something in there for people to take home with them. And with that, I'll close and hand over to Tina, who's going to take some questions, I think. Thank you.
Matt, is AMR the next pandemic?
Well, I think I kind of alluded a little towards that in the talk, and mention of AMR being a silent pandemic. I mean, it is a pandemic, and it's a pandemic that requires more attention. I've mentioned some of the attention it's getting at the sort of governmental and international level, but I think there is certainly more that needs to be done. I think there are things that we've learned from the current pandemic about the way maybe we develop new therapies, vaccines, and get those accelerated. I've spoken about clinical development pathways and regulatory acceptance of drugs that are being developed, and I think that whole process has been improved, and hopefully, I think new antibiotics can progress through that system much faster, given some of the innovations of the last year. So yes, it's a pandemic, and I think we're learning more how to deal with it as a pandemic.
You use natural products to develop new antibiotics. Is it possible to develop antibiotics purely synthetically? And if so, is there potentially limitless supply of new antibiotics that we have yet to develop?
Yes, and I guess in my kind of last slide, I was suggesting maybe that we want to use artificial intelligence to design new antibiotics, and the first completely artificially designed drug has recently been approved for use. That's a drug for use in cancer. So I think those sorts of technologies do have a place, and I think they may lead to new antibiotics. But I'll also, as a word of caution, really, in a way, refer back to the diagram where I had the golden period of the golden age of discovery and the discovery void. And in the middle, we spent a lot of time and huge amounts of money using new approaches, which included kind of synthetic approaches to develop new antibiotics. And that had a lot of promise early on. So you can quite easily find something that looks like it'd be a really good antibiotic. It will interfere with the way bacteria grow and replicate. But when you try and use that as a drug, it won't get into the bacterial cells, or some other reason it's not going to be a good drug. So for synthetic development of antibiotics, I think that's a kind of a word of caution really there in a way. I think some of the newer technologies and approaches that are coming into use now will help. But I still think there's just such an immense diversity of natural products that they still very much warrant investigation. Yeah.
What are your thoughts on the identification of novel bioactive agents from natural sources and the direction of travel in future therapeutics regarding COP26 and a more three-dimensional approach to sustainability in pharmaceutics that is ecologically friendly?
Wow. Right, okay. COP26. Well, in our defence on the sustainability front, I mean, when we talk to people sometimes about the idea of going to the deep sea and harvesting these sponges and looking in them for new antibiotics, you can see people thinking, oh, you're going to go and strip the seafloor of these amazing animals and extract antibiotics from them. But because we're looking at the bacteria that grow in these samples, we can isolate the bacteria, grow them up, and then produce the antibiotics that way. So we may only need one sponge to find the next new antibiotic. So some of these approaches are very sustainable for that sort of reason. And I think maybe staying on the kind of the topic of sort of sustainability and climate change, those sorts of things. I mean, we need to understand the marine ecology because there's clearly a vast untapped biodiversity in these deep ocean spaces. And we don't know what the impact of climate change is going to be on those spaces. So is that going to stop some of these sponges being able to grow? Are deep sea mining technologies and approaches going to destroy some of this potential biodiversity before we see it? So I think some of the work we're doing, and I think this is what Kerry was kind of suggesting earlier, maybe that hopefully we can kind of elevate the awareness of the potential benefits, health benefits, human benefits from the deep sea environment through some of this sort of work. So I think maybe that addresses parts of that question, but maybe not all of it.
Someone's interested to hear comments about global trends with AMR and is wondering whether your future research will get you back to New Zealand.
Well, actually, so I came back from New Zealand in 2001, and I didn't mention that I've had 3 trips back to New Zealand to work in John Tagg's lab, and I've got some papers from those subsequent trips. There's nothing particularly lined up at the moment for a return visit to New Zealand, but John, if you're still listening, I know he's still active in research in Dunedin. I'd be very willing to collaborate with him again. Yeah.
What action should NHS trusts take to tackle the issue of AMR?
I think NHS trusts and I think GPs, medical practitioners, pharmacists, I think they're all very aware of some of these sorts of messages that are being used to promote the risks of AMR, and clearly talking to the clinicians in Brighton earlier, that they're very aware. They do very regular audits of the antibiotics that they're using. They are very careful about cycling antibiotics around to try and minimize the development of resistance. So I think really in clinical settings, it's known to be a major problem and is being addressed in as best a way as possible. So I think the problems probably lie elsewhere, and a lot of the solutions probably lie elsewhere in things like agriculture and development of vaccines to reduce the amount of antibiotic use in agriculture. And I think probably time for one more question.
Really nice last question. With regards to Alexander Fleming, how do you view impacts of chance encounters, luck and resilience to stick at what you love in research?
Yeah, I think there's a few key things there really in a way. I mean, sticking at what you love in research, I've been fascinated by everything that I've been able to do. I'm probably fascinated by too many things. That means I get involved in too many different things rather than focusing completely on one thing. So I think having a real passion for the work that you do is key. Certainly, if you're entering into this as a kind of research career. Sorry, the first bit about Alexander Fleming.
How do you view? With regards to Alexander Fleming, how do you view impact of chance encounters, luck and resilience to enable you to stick at what you do? Yeah, and chance encounters. I mean, the kind of introduction. When I came to Plymouth, I really wanted to meet and start working with marine biologists. I didn't have this sort of work in my mind when I was keen to do that. And it was a chance encounter that led to the introduction of myself and Kerry. And that's led, as you've heard, to a number of PhD students and other things. So, yeah, I would say that chance encounters are important, but it's what you do with those encounters as well that's very important. And one thing I didn't really emphasize very much was kind of collaboration and working with people. This is the sort of science, and you mentioned earlier the need to work across disciplines, but you've got to be able to work with those people. And so I think key is developing relationships with people that you're working with that you know are going to lead somewhere and are going to be beneficial to both parties. So I think collaboration is essential, but it's also essential to make sure those collaborations work in two directions and are beneficial. So be very careful if you're venturing out in this sort of field, but very careful about the collaborations you make and the relationships you develop, because that's key to the way you'll progress. Thank you, Matt. Have you got any final comments?
No final comments other than just a thank you again to everybody for tuning in and coming along. I think we often say to PhD students when they head into their Viva, and this was probably said to Poppy yesterday, enjoy it. It's about the only time you'll have to kind of sit and talk to somebody for 2 or more hours about your research. I don't think PhD Vivas are often enjoyed in that sense as much as we suggest, some shaking heads in the audience. And for me, I think partly preparing for and delivering this talk in a little way has been an indulgence. So equally, you don't have many opportunities to do what I've just done and kind of talk about your own research, whatever you want about your own research to an audience. So thank you for allowing me that indulgence. And I hope you've all taken something away. Thank you.